Okay, welcome everyone. I'm Sherry Hume. I'm a co-founder of Cure JDM Foundation, and I have the honor of introducing our four speakers today. We have Dr. Ryder, who is a pediatric rheumatologist and a consultant attending physician and clinical professor of medicine at the George Washington University Myositis Center, which is one of our funded centers. Cure JDM funded. She's also the Deputy Chief of the Environmental Autoimmunity Group at the National Institute of Environmental Health Sciences at the NIH. Also, we have Dr. Patricia Dranchak. Did I say it correctly? Okay. She is a senior research scientist at the Division of Preclinical Innovation at the National Center for Advancing Translational Sciences, also at the NIH. We have Dr. Nagaraju, which we call him Raju, or Dr. Raju for short. He is the founding chair and professor, Pharmaceutical Sciences, Binghamton University, State University of New York, co-founder of 2 biotech companies. And you guys are really lucky, by the way, because I'm doing the very shortened version of their bios. Dr. Agarwal, did I say that right? Okay. He is an associate professor of medicine, Division of Rheumatology and Clinical Immunology at the University of Pittsburgh. He is also the medical director of the Arthritis and Autoimmunity Center and co-director of the Myositis Center at the University of Pittsburgh. So please join me in welcoming our speakers. And Dr. Ryder is going to start out.
So I'm going to start with a general introduction for this session on drug discovery and emerging treatments, kind of giving you an overview first about what are clinical trials and how do they work and how new agents are approved and tested. So most studies start with phase 1 studies that are first in human use and just established a dosing in a small group of healthy volunteers. And these are typically fairly small studies of under 100 patients. Sometimes they're only 10 patients big. And the success rate at this stage is about 70%, sometimes less. Then if something is successful at phase 1 and a company wanted to go on, then they could evaluate that efficacy and safety of a drug or a biologic treatment in a larger group of patients with a particular disease. These are typically still small studies. They might be anywhere from 50 to several hundred patients big. Typically, if a company wants to license a new treatment for a fairly large disease like rheumatoid arthritis or high blood pressure, they'd have to do 2 of these phase 2 studies. Whereas if you have an orphan disease like myositis, you might only have to do 1 phase 2 study. Even a good phase 2 study might be enough to get a license application. So these studies also aren't always successful, but they also often testing different doses of a treatment, especially in that population, because we don't always know the optimal dose and optimal outcome to look at. So this is a more exploratory type of study. So this is also about 33% success rate at this stage. And then we get into what's called a pivotal study or phase 3 study that really confirms the drug's efficacy, monitors side effects in a larger group of patients. Typically, these are several hundred to several thousand patients that are needed before a new treatment could be licensed by the FDA. Usually, they're conducted in multiple centers. Usually, they take a few years. In fact, we're talking from phase 1 through phase 3. It can be more than a decade. In these studies, I put down 25 to 30% success rate. That's what I found online. But frankly, all the way through to licensure are less than 5% of treatments that go from phase 1 all the way to license. And then phase 4 are studies that are conducted after FDA approval in a post-marketing phase. And these are trying to evaluate longer-term side effects in larger populations. So it's very important for people to volunteer for trials. Actually, some of these slides are from Sherry Hume last year. With her help, we did a session last year. Many trials take a long time and millions of dollars, as you heard this morning. And it could have a great idea, but it doesn't mean that it's going to test out and really make it all the way to licensure. But actually, the lack of participants in studies is one of the main reasons that trials are not finished. In fact, I'll just tell you, we had a small treatment trial of intralesional sodium thiosulfate for calcinosis being conducted by Dr. Allison Ehrlich in GW. We only had 2 patients enrolled, so we had to publish with 2 patients. So, and it took several years to do that much work. So it's really important for people to participate. Okay, so we have challenges in myositis, and particularly in juvenile myositis, because these are very small populations, very rare diseases. The diseases are very heterogeneous, meaning that not all the patients are alike, and often not well understood, and they have a fluctuating up and down to them. There's often a lack of interest from big pharmaceutical companies because these are not going to be blockbuster money makers for them in such a rare population. So we often have little evidence and a small number of patients to study. We also have, really frankly, relatively few sites and investigators that could do these trials, but we have more these days than we did even 1 or 2 decades ago, because we have now a lot of international collaboration, a lot of validated assessment tools, and new response criteria. In fact, Dr. Agarwal and I work together closely on the new response criteria, which are now approved by American College of Rheumatology and European League Against Rheumatism. And then it's difficult for patients to enter a trial. Their disease has to be active enough. They have to sometimes decrease the amount of steroid or other treatment that they're on because we're worried about side effects of too much treatment, and the treatment has to remain steady during the trial. Nowadays, we have the added complication that we're giving patients rituximab more and more as a 2nd and 3rd line treatment. And because that one is knocking out the B cells for a while, we can't often give another immunosuppressive medicine right on top of that. So we have to wait a while till we could give something new, like Orencia, for example. People also can't have too much damage or scarring in their disease, because then we won't be able to see how much improvement there is. Their disease really has to be in a more active state when they're included in a clinical trial. So not all patients are good candidates for clinical trials. We are fortunate that we have some legislation that is stimulating pharmaceutical development. One of them is pediatric legislation that came about from the FDA and the European Medicine Agency that are encouraging companies to do more development and get more licensure of treatments for pediatric use. So we have the Best Pharmaceuticals for Children Act, but this is a voluntary situation. The timing of when the FDA wants companies to test things in pediatrics is end of phase 2, because they want to establish general safety of the treatment in adults first. But they're giving some incentives or rewards for companies that will go forward with this. For example, they'll say that if you do the studies, then they'll give 6 months of market exclusivity on the drug or biologic, which gives companies financial incentive to do this. It covers all drugs and most biologics. And orphan diseases can be included, and that FDA, when it is an orphan disease, they'll even make a written request for companies to try to do it. And the goal is to get more pediatric specific labeling, but it is voluntary, so not all companies take this up. In Europe, we're a little bit more fortunate, and we've seen more things being pushed from Europe that are impacting the US actually in the last few years. So that they're mandating that pediatric studies be done. They want a plan at the end of phase 1. It doesn't mean that it has to go into kids at the end of phase 1, but they want the company to make a commitment at that time. And they get 6 months of patent extension if they do the studies, and they include everything, drugs and biologics. And orphan diseases are included. And they have a broader review outside of the agency. A pediatric committee reviews the studies and approves the license. So we're seeing things push from Europe, and sometimes when they're international, we get things done in the US too. Then we have a number of benefits through the orphan drug programs, programs for very rare diseases that encompass less than 200,000 people in the US. If you get orphan drug designation, then you have 7 years of exclusivity or no competition. You can get the company will get a tax credit of the clinical trial testing costs, a portion of that, and a waiver of their drug application fee. And then there's something called Rare Pediatric Disease Priority Review Voucher. So if they're studying a rare pediatric disease, they get a priority review for another indication for a different product. There's also Orphan Products Grant Program. So some studies are getting done through this type of grants, and there are even new natural history grants as well, a fairly new grant within the Orphan Products Program. And then there's something called FastTrack or expedited programs for serious conditions. This is used a lot for cancer, for example, where you try to get things more quickly to market because there are no effective treatments and it might really be life-saving for patients. So there are different types of FastTrack and all these things. If you go for one of these type of designations, then you get a lot of guidance and expedited review from the agency. Priority review and things. And I think you'll hear from Dr. Nagaraju that they're trying this approach for one of the new treatments for accelerated approval, meaning that there's a meaningful advantage over available therapies. And in this case, you don't have to show necessarily that the treatment was clinically effective, but you could use surrogate endpoints, other types of endpoints that would predict that there's clinical benefit, and then later do confirmatory trials to show the benefit. So that would get things to market more quickly when you use that approach. So with these orphan incentives, then we're seeing a lot of development in orphan products in the last few years. So that back in the 1990s, we rarely had orphan drugs approved, but now in 2015, almost half of the drugs approved by the FDA were for orphan or rare diseases. The positive side is that we're getting a lot of interest in rare diseases. We're having insurance coverage for these medications and many new available treatments that were much needed. But you will also read about that some of these medicines for these rare diseases can be astronomically expensive. So that's one negative. But I would say with all these pediatric regulations, the orphan regulations, and the fact that we have gotten a lot more organized in the myositis world with better endpoints, a lot more international collaboration, and better targets for treatment, that we're seeing a lot more pharmaceutical interest in myositis than we had seen previously. So there are a lot of benefits to volunteering for a clinical trial. One is that you can get potential access to treatment that may not be available outside the trial. So you might get a really new treatment that's kind of exciting. This treatment could be more effective or even safer than standardly available treatments, and you would be the first to benefit if you were in the trial. And you will be closely monitored by researchers, and you'll have additional testing. You'll have chance to see doctors fairly frequently and learn more about your disease. And your actual involvement will help move research forward. There are risks to being involved in a trial. The treatment may not be any better than standard treatment. It may have unexpected side effects. You may need additional visits to the doctor, additional testing, and you may not benefit from the treatment. So I'm just going to go over here. We talked this morning. I'm not going to go in great detail, but we currently have 4 trials for JDM that are currently enrolling patients, just to be aware of. One is the abatacept trial in JDM at George Washington University Myositis Center. Everybody in this trial is getting the treatment. It's what we call an open label trial. It's an injection, a subcutaneous treatment. Patients have to have moderately active disease and have failed prednisone and one other treatment, and they have to be at least 7 years of age. And you've all received a handout in your packages with information about the trial. We also have Aksar Gel. I'll go into that one in a minute because I haven't discussed that one before. I also spoke about this compassionate use study for baricitinib. This is the JAK kinase inhibitor. It's being tried in severe juvenile dermatomyositis. It's an oral drug. It inhibits that interferon pathway that we spoke about this morning. And because it's a compassionate use study, the company just was approved for license for rheumatoid arthritis. So this program is going to close at the end of the summer. So it's a very short window for JDM patients to receive this treatment. I mean, once you're in the program, you'll receive it for a while, but you have to get in the program by the end of the summer. That's how long it will be available. The last one is sodium thiosulfate for treating calcinosis. This is for patients with moderate to severe calcinosis. It's an IV treatment. It's many visits to the NIH, and one of those visits, it's a very long visit for 10 weeks where you get IV treatment 3 times a week. And Dr. Schiffenbauer is running that study. Again, that was in your packets here at the conference. So I'll just speak a minute more about the Aksar Gel trial, which is being run out of the Cleveland Clinic. Actually, Dr. Agarwal, who's going to present next, has done studies of Aksar Gel in adult dermato and polymyositis, and he may even speak about that with us. But this is adrenocortical steroid hormone, and it's like a steroid or corticosteroid, but it has some other properties, including pro-opioid melanocortin peptides that may also downregulate the immune system. So this one is open for patients who are adult age, actually, with JDM or adult dermatomyositis or have clinically amyopathic disease. And it's trying to study the benefits on the skin disease. And it is a medicine that's given by injection twice weekly. So that one is available at the Cleveland Clinic. Dr. Agarwal is going to speak with you about there are many adult studies available, and they're very interesting to learn about because someday those may be available to JDM patients. But these 3 studies are available to adults who have JDM, and one is a very large controlled trial of abatacept or Orencia. One is tocilizumab that Dr. Agarwal is running in Pittsburgh, and there are multiple centers. And one is for a medication for adult dermatomyositis, but again, adults with JDM can enroll, and it's blocking interferon beta. And I think Dr. Agarwal will give you a little bit more about each of these studies. One of the things to be aware of, keep your eyes on, are treatments that get approved for juvenile idiopathic arthritis, because these treatments, there are more treatments approved for JIA than JDM, because JIA is much more common than JDM, even though it is also a rare disease. But it gets studied often because a lot of treatments get approved for adult rheumatoid arthritis, and then they're mandated to study it in children because that's the pediatric equivalent disease. So treatments get approved, and one advantage is that sometimes JDM patients can be prescribed these treatments if they have a lot of arthritis and their doctor says they have arthritis. Then that's a way to fool the insurance company and sometimes get these medicines, so to speak, for your child with JDM. So some of the treatments, and also then some of these treatments go on to be studied also in myositis patients. So it's just good to know about these treatments. The other advantage is that they've been then studied well in children. You know, the dosing is worked out, and that also has some advantages. So these, I think, are things for us to keep our eyes on in terms of future treatments for JDM patients too. So we have a number of treatments, including anti-TNF alpha blocking therapies. As it turns out, these haven't been as successful for myositis as we had hoped. It's not usually in JIA, it's a very key treatment. In JDM, we don't know if it's very effective, and we usually use it much later as a more of an experimental type of treatment. There are also IL-1 blocking therapies, another cytokine that gets blocked, a pro-inflammatory cytokine, and then tocilizumab that Dr. Agarwal will speak about, and abatacept are already approved for JIA. So rituximab also has been approved, actually is being studied, as well as tofacitinib, a JAK kinase inhibitor, and belimumab, which is anti-BLyS, which blocks B cells. So you can keep your eyes out on trials that are available through clinicaltrials.gov, which is a government public source website to learn about different clinical studies. You can use the search terms juvenile dermatomyositis or dermatomyositis. Some of the studies in clinicaltrials.gov are not treatment studies, but just observational studies. So you will also see, for example, our studies from NIEHS that are looking at environmental and genetic factors are also listed in clinicaltrials.gov. And again, you could volunteer if it's appropriate, and you in this room could even encourage other patients to volunteer and be supportive to them. I think that's very helpful. Okay, I guess we'll take questions at the end. So, okay.
Thank you for everybody being here and giving me this opportunity to present this talk. So my job is to talk about adult dermatomyositis clinical trial, which has a lot of implication for juvenile dermatomyositis, because a lot of time the drugs are first tested in adults and then they can be tested in pediatric population, and a lot of time the drugs that didn't work in adults, most likely they may not even work in JDM patients. Another thing is safety and tolerability. So you can learn a lot about safety and tolerability in adults. So rather than having kids being exposed to those safety and tolerability problems, you can weed out all the issues in adults and then apply in peds. So to me, actually, this works out really well that, try a drug in adult disease first and then, move it to the juvenile disease. To me, this strategy is really good. Now, there are adult patient trials which are actually enrolling JDM patients as well, the adult JDM patients, the patients who are beyond 18 years of age, but the diagnosis was juvenile dermatomyositis. So here are some disclosures. And the thing is, if you probably look from my disclosure, is there are many companies that are interested in clinical trials in myositis now. So I'm working with many of them, and I've worked with almost all of them saying, okay, if you want to do a trial in our population in myositis, we'll help you. We'll help you do a trial, whichever way you want to design in JDM, polymyositis, dermatomyositis. So we are out there to help them to design these trials, because you have to understand from the company standpoint, they don't have the expertise in these rare diseases. So they need to have a partnership with academic researchers like us to really come up with a good design, because if their study fails, it's not their study failed, we failed. Our patients failed, right? So it works out both ways. We want their help as well. And these are the companies who comes up with a novel molecule to think about. So we really need to help them to come to the forefront and do trials in our diseases. So the other thing is that if you look at the landscape of all the clinical trials that are currently done, most of the clinical trials are being done in adult dermatomyositis patients. Some of them are being done in juvenile dermatomyositis, fewer, I would say, in polymyositis. So, but most of the clinical trials are currently going for adult dermatomyositis. And we want to encourage to do trials in multiple subsets of myositis. So, for example, trials doing both adult DM and adult PM. But a lot of time there are issues that the drug may have more preclinical benefits seen in dermatomyositis, or other studies, safety studies have been done more in dermatomyositis. So FDA says, wait a minute, why are you doing in two diseases? Just do it in one disease and then go to the other disease. And sometimes FDA allows us to do PM and DM together. So I've been in conversation indirectly with FDA, and every time we go to FDA, we get a different answer. And that's because they don't really have that much experience in these diseases. So it depends on who's reviewing the study at that point. So we get different answers. Sometimes they say, okay, combine the two. Sometimes they say, no, you have to do DM alone, then do PM. So it's really confusing even for us. So you will see in subsequent slides, there are many studies which are DM and PM combined. Some are only DM and so on. The other good thing that has happened in rheumatology in general is that because of rheumatoid arthritis, lupus, and other common diseases, we have gotten a lot of molecular targets. By molecular targets, what I mean is there are multiple immune pathways for different diseases. So for rheumatoid arthritis, may have 4 or 5 different active immune pathways. For this is a pathway that we put together for myositis. And if you can see, there are within this diagram also, there are 4 or 5 different pathways through which the inflammation can come to the patient muscle and skin. So the good news is that all of these inflammatory pathways overlap with each other. So whatever we learn from rheumatoid arthritis can be applied with to juvenile dermatomyositis or dermatomyositis or polymyositis. And because more and more molecular pathways are being designed, more and more molecular targets are now being coming up. So how to block this pathway or that pathway or that pathway. So these companies are coming up with drugs to block these pathways at different stages. And it depends on how you study them, but many of the drugs then can be applied to rheumatoid arthritis. We learn from them and then can be applied to our diseases because of these overlapping inflammatory pathways. So the first one, I'm going to talk about this drug, abatacept. There are about 3 or 4 studies currently going on this, so it's really important one to know. So what it does is inhibits a T cell co-stimulation. As you may or may not know, there are 2 types of major immune cells called B cell and T cell. And these are the immune cells that go and attack our muscles and skin, cause inflammation. So what this drug does is it prevents the activation of one type of immune cell called T cell. By doing so, it kind of downregulates the inflammation in somebody's skin or muscle or joints and so on. So in this, the other good news about this drug is this has been approved for rheumatoid arthritis for last 1 decade, more than 1 decade. It's also approved for juvenile inflammatory arthritis, which is JIA. The other things that are very encouraging about this drug, that I actually am studying this drug in an interstitial lung disease patients. That's because in rheumatoid arthritis, this drug has been shown that if you give this drug to rheumatoid arthritis patient with ILD, their ILD doesn't get worse. Whereas if you give other biological drugs, their interstitial lung disease or ILD gets worse. So to me, we used that to our advantage and said, why don't we use this in ILD, which is the pulmonary fibrosis, in myositis. So we're now trying to study this drug in patients with interstitial lung disease and myositis, gaining the experience from the rheumatoid arthritis literature. Also, the other thing, if you look at the basic science, a lot of myositis pathways and myositis interstitial lung disease pathways are also T cell mediated process. So this blocks the T cell activation, so kind of makes sense biologically as well. And there are case reported series in which it appears that when patients were given myositis patients were given these drugs, abatacept, because these drugs are commercially available. So somebody can label somebody as rheumatoid arthritis because they have myositis, but they also have arthritis. So if they are labeled as rheumatoid arthritis and they get these drugs, their myositis gets better. So some of these case reports have come out that this drug may actually help. And it also is one of the safest drug in rheumatoid arthritis. In general, we hear biological drugs are bad because they cause immune suppression and cause horrible infection. But this is one of the best biologic drug in rheumatoid arthritis world that doesn't cause that much infection. So because of that, Dr. Lundberg from Sweden did this really excellent study in which it was basically in this study everybody got drugs. Half patients got drug early and half patients got drug after 3 months of delay. And what the results were, the patients who got drug early got better more and earlier, which I will show you on the next slide. It's more clear based on a new ACR-ULA response criteria. If you look at the black lines are the patients who got drug early. They got better faster and then sustained it. But the patients who got drug late at 3 months, then they got better after 3 months. So really teasing out between the drug early and drug late. This trial then led to one of the major trials that are currently ongoing. This is a phase 3 randomized controlled trial which the drug company BMS is doing. 55 sites, they're doing international study. This is US, Australia, Asia, and South America. The target enrollment is 150. I think they have enrolled around 20-25 patients so far. I'm in constant communication with this company. And what they're really studying is efficacy and safety of abatacept, a sub-Q injection of this drug in patients with idiopathic inflammatory myopathy, which basically, if you look at the inclusion criteria, includes polymyositis, dermatomyositis, necrotizing myopathy, overlap myositis, but also juvenile myositis. This is one of the major trials that actually does include patients with juvenile myositis, juvenile dermatomyositis, who are older than 18 years of age. You have to be older than 18 years of age. So they're using the previous outcome criteria, which is IMAX definition of improvement, because when the trial was started, at that time, we didn't have the new response criteria. So they're using the older response criteria, and this study is ongoing. Then we have abatacept in juvenile dermatomyositis, which is only 10 patient study, but it's an open label single arm study. So it's a small study at GWU, and currently is enrolling. Do you know how many patients, Lisa? 4 have enrolled. So I'll encourage you to enroll in this study to think about, because especially because this study is this is the same drug being studied in adult population, already has one proven study. Second major study is ongoing. If that study is positive, this will become an FDA application for approval. And then another study that we are doing that I talked to you earlier about, abatacept in interstitial lung disease in patients with dermatomyositis and polymyositis. So this study is, you got to have interstitial lung disease, and you got to have this antibody called antisynthetase antibody, which is the most common of antisynthetase antibodies, Jo1 antibody. So if you have the antibody and you have the lung disease, then you can enroll in this study. This is a smaller study, only 20 patients, but it's a placebo-controlled, randomized, double-blinded study, which we have enrolled 3 patients so far. We have 5 centers in US, and it's ongoing. So essentially, there are 4 clinical trials on abatacept, 1 completed positive, and then 3 are ongoing on this drug. So we're really hoping that this drug comes through in other trials as it came through in the first one. So shifting gear to another drug called tocilizumab. This is a drug that inhibits an cytokine called IL-6. IL-6 is in general pro-inflammatory cytokine, which means basically it promotes inflammation. So blocking this cytokine, we hope that it will cut down the inflammation in muscle and skin. And there are mouse models in which they showed that when you block the IL-6 pathway of inflammation, it actually improves the myositis in the mouse. Obviously, it's a mouse model. We don't know in humans, but at least that was encouraging. And there are many other basic science or preclinical studies which suggest that IL-6 may be a major player in myositis. For example, it's overexpressed, which means it's seen in abundance in patients' blood and muscle of myositis patients as compared to non-myositis patients. So there are some basic understanding that makes us think that this drug may work. So based on that, we are currently enrolling for this phase 2 randomized double-blind placebo-controlled trial, which is for 6 months, and this is IV infusion, although this drug is also available as an injection, subcutaneous injection, but right now it's IV infusion. You're going to get drug every month for 6 months. And in this, we also are enrolling juvenile dermatomyositis patients who are above 18 years of age, along with PM and DM. We have enrolled so far 36 patients out of 40, and I think we are on the last couple of months of enrollment. And this study, although only 40 patients, we had significant difficulty in recruitment. We have 7 centers for the study, but it's still difficult to recruit. That's what I keep telling people, that it's very difficult to recruit because it's just in general patients don't want to go for the studies because it's cumbersome, it's time consuming for them. But if they don't come for the study, how are we going to provide them better drugs? So it's kind of difficult situation for us, but it is what it is. So we have it is true for all the rare diseases. It's very difficult to recruit for these studies. Another novel drug I want to talk to you about is this drug called Lyna Bassam. I don't know how to pronounce Lyna Bassam, and it's a novel synthetic oral molecule. It's an endocannabinoid mimetic drug, which I will explain in a minute. But basically what it does is, if you think about this way, what happens is if you have an injury, our immune cells goes to the injury, try to repair the injury, cause inflammation there in order to repair the injury. In dermatomyositis or juvenile dermatomyositis, what happens is immune cell goes to our muscle or skin without an injury. They basically try to attack the muscle and skin, the immune cells, leading to inflammation in those organs, right? So in general, in normal healthy being, what would happen is if there is an injury and immune cells goes there to cause inflammation to repair the injury, what happens is there's a shutdown of that inflammation, which we call as resolution of inflammation. In a normal healthy being, that would happen, and that inflammation shuts down and your wound or injury would heal up, okay? But in abnormal situation, in the disease situation, what happens, that resolution of inflammation doesn't happen. And that's why that inflammation continues to happen. One, it's a target is wrong, means muscle and skin which were not even injured, the immune cells go and attack those organs. But the second thing, there is no resolution of that inflammation. The resolutory part is absent. So what this drug does is actually blocks with certain receptors on the immune cells, then that promotes that resolution of inflammation. So essentially, it's not like anti-inflammatory. Most of our drugs are anti-inflammatory. We want to prevent inflammation. We want to prevent from immune cells from going to attack the organs or from expanding and doubling up and tripling up. We want to, that's called anti-inflammatory or immune suppressive. This is an immune suppressive. This is okay. You cause the inflammation. This is going to shut down the inflammation through a resolution route. So it's a very different drug and has been shown promising results in preclinical models of inflammation and fibrosis. And I'm showing you one of the slides from Dr. Worth, which shows really the CDASI is the skin disease activity in dermatomyositis patients. And if you see, this is the normal, up until this was a normal standard of care treatment, and once they got this drug, their disease activity, the skin disease activity, dropped down rather quickly. So it's some promising results in the study, preclinical and now clinical, that this study is now the drug is now going into major phase 2 clinical trial in dermatomyositis. I don't know if they're including juvenile dermatomyositis or not. It's not yet active, and we are constantly working with them. But it's also, they're also doing studies in cutaneous scleroderma and cystic fibrosis. So this is their study that they are planning. They're trying to do safety, tolerability, and efficacy of linacimab in adult patients with skin predominant dermatomyositis. That means they have to have skin disease to begin with. This is a double-blind randomized placebo-controlled trial. 30 patients. Dr. Wirtz is a PI of the trial, and the primary outcome is currently being debated. Should it be the new myositis response criteria or should it be the skin disease criteria? And there's still ongoing discussion going on right now. Then shifting gear to a really old drug called IVIG. You may have heard about this drug or even used this drug. This is a drug, if you think about what year is it, 1993, the first clinical trial done in myositis, one of the only clinical trials that was really successful. In this clinical trial, half the patient got IVIG for 3 months, then half of the patient got got placebo for 3 months. Then they switched. So the IVIG group after 3 months got placebo, and placebo group after 3 months got IVIG. It's really one of the remarkable studies done. We learned a lot from this study, but the bottom line is the results were really good. So this is the patients who got immunoglobulin or IVIG first. They all got better. If you can see that most of the patients got better. When they got placebo, either they stabilized or got worse, means they flared up after stopping the drug. Whereas the patients in the placebo either didn't get better or were stable. And then once they got IVIG, some of them got better. So it's really remarkable results. And since then, we have been sporadically or sometimes higher frequency, sometimes lower frequency, being using this drug in our clinical care. The problem is this drug is not FDA approved. So what happens when I try to give to patient, it really depends on the insurance company whether they're going to approve this drug or not. So it's so much insurance dependent that, because it's not FDA approved. So now companies are interested in this drug, and the companies who are actually developing IVIG saying, why don't we do a study and prove it once and all if it's going to work or not. So because of that, there's a major study going on IVIG in refractory dermatomyositis, and I'm heavily involved with this study. This is double-blind randomized placebo-controlled phase 3 study in which patients will get IVIG or placebo for 4 months, 0 to 16 weeks. Then everybody will get the drug for another till 40 weeks. And the response criteria is new ACR-EULAR response criteria, which is total improvement score. We are also measuring skin and other things. But I think this study is really critical study, and even they are having trouble enrolling patients. Significant trouble in enrolling patients in US. So far, they only enroll, I think, 20 to 24 patients in US, but most of their patients are coming from Europe. This is a drug we believe that it works. So even that drug, we are having difficulty in enrolling. So think about that, you know. So and then couple of other good news is that IVIG also comes in sub-Q IG form. The same drug can also be given subcutaneously. Previously, it was very difficult to give the volume of IVIG that could be easily given through IV through a subcutaneous or an injection in the skin. Now they have developed technology that it can be concentrated and given through the skin easily. The advantage is if you give IVIG, you have to give every month. Your patient has to come in, get infused for several hours, sometimes up to 8 hours in an infusion center. So it loss of work, and then people can have headaches after it. So another loss of work. So they're developing sub-Q IG where basically you can get injections twice a week at home. You can give a sort of an injection yourself twice a week at home, so you don't lose work day. So this same company is we are trying to do now an international study with sub-Q IG. And the way the study is designed is that everybody comes in with the good disease activity, means with remission, means they are doing well after they have gotten IVIG. And then they're going to do a placebo-controlled trial to half a patient will get the sub-Q injection and half will get placebo to see if we are able to maintain people in a good disease activity after they have gotten better with IVIG. Because another problem with IVIG, how long do we continue IVIG? 6 month patient is better. Now what should I do? Should I stop it? If I stop it, they get worse. So the idea is if they could go to a simpler regimen, then that would be more sustaining for another year or two. So that's the drug and another company is interested in doing sub-Q IG study as well. In the interest of time, I'm going to quickly talk about tofacitinib. Is a drug. It's a JAK kinase inhibitor. It's a little complicated mechanism, but basically it's an approved drug in rheumatoid arthritis. It has changed the landscape of rheumatoid arthritis in last several years because they used to be all oral drugs in rheumatoid arthritis. We used to call them as DMARDs or disease modifying agent. Then the era came of biological drugs, where all the drugs were IV or sub-Q, and for last 10 years, that's all we have seen. Now the things are shifting to small molecules. The advantage of small molecules is it's an oral drug. So now rheumatoid arthritis landscape has changed again in the last few years. We're now prescribing back oral drugs after the approval of tofacitinib, which is the JAK kinase inhibitor drug. So the same drug is being studied in patients with dermatomyositis, and through Hopkins, 30 patients to be enrolled. I think they were also having difficulty in enrollment. I think it's only adult DM. It's only adult DM. Okay. Only adult DM. Yeah. No, adult JDM is not allowed. Okay. They are also having a little difficulty in enrollment, if I understand. So even an oral drug, again, difficulty in enrollment. That's the theme we hear all again and again. And then there's an interferon beta blocker or anti-interferon beta drug that is being currently enrolling patients. These are the adult patients with dermatomyositis, but they're also enrolling adult JDM patients. So this is a really good study to think about. And the reason this is a good study, because if you look at the basic science, if you study the patients with juvenile dermatomyositis and dermatomyositis, interferon is a gene signal which is really upregulated in patients with dermatomyositis and juvenile dermatomyositis. So any drug that is blocking the interferon signal may have a good efficacy. So this is a drug, this is an international study. No, it's not an international study. I think this is a mainly US study with only 30 patients, but they have about 10, 11 centers that are currently active. They are enrolling DM and JDM patients. And some of the studies in the interest of time, I didn't put in the more details, but belimumab in myositis, belimumab is anti-BLyS, but what it essentially does is blocks the B cell. I told you there are two types of immune cell, B and T cell. This blocks the B cell. The reason to block the B cell is because although when we do a biopsy of dermatomyositis or myositis patient, polymyositis patient, we mostly see T cell, but we do see some B cell. And we believe these B cells are helping the T cells to cause a disease. So there's sort of a supply chain for the T cell where they give them cytokines and chemokines, sort of molecules to help T cells to keep going. And that's why blocking B cells can be sometimes as important as blocking T cells. So this is a drug that blocks B cell through indirect way. Aksar has already been discussed by Dr. Ryder in refractory cutaneous dermatomyositis patient, and that does include DM, JDM, and clinically amyopathic dermatomyositis. Also, apremilast, which is a cutaneous recalcitrant dermatomyositis treatment. This is only 10 patients open label study. This drug is approved for psoriatic arthritis. Again, you're seeing the same thing. Drugs approved in other diseases are being applied here more and more. That's why there's so many drugs nowadays that can be studied in myositis. Interferon alpha-quinoid, a study from France is being done in Europe. And sodium thiosulfate for treatment of calcinosis associated with JDM and adult DM is done by Dr. Ryder. And I was a little surprised to see the N of 250, which is really, is it not true? 18. So you got to change it on the clinicaltrial.gov, because I was like really surprised. I've never seen, the largest trial ever done was the rituximab trial. It took us 10 years to enroll 200 patients. So I don't know where you're going to get those 250 patients, but that is what is on the clinicaltrial.gov. So the other good news I want to give, and I can't share too much details about this part because company specifically told me not to. But if you remember, I was telling you the interferon signals are very high. These are the genes that are upregulated, means they're active in patients with dermatomyositis. So if we compare patients with dermatomyositis and normal healthy patients or other immune disease patients, these genes are really activated. So the idea is if we could somehow block the action of these genes, we could reverse some of the problems that we see in dermatomyositis. So those are called anti-interferon treatment or anti-interferon drugs. And I'm currently in conversation with one of the company who is ready to launch it in the go to the phase 3 clinical trial with this drug. They have a really good drug that they showed in a lupus at least that it really worked well. So now they're concentrating on the other diseases. And lupus is another disease where the interferon signal is very high. So dermatomyositis and in lupus are two diseases where interferon signal is really high. So blocking that would be very helpful. So I'm really hoping this drug comes into the clinical trial rather soon. Thank you very much.
So far what we have been doing is we are simply borrowing drugs that worked in other diseases and trying to see if they'll work here. So there is so much you can borrow because it's very hard. CureJM, those of you who have looked at the strategic planning, is very clear in their vision. That is to develop a pipeline of drugs for cure juvenile myositis. How do you do that? That means you have to go back to basics, understanding the disease, coming up with pathways that are unique, and if we come up unique pathways, how to develop the drug from the start. So we have to take both approaches. We simply cannot go and start developing drugs. It may take 20 years. So this is the right way. I think I want to thank Sherry and the leadership team for having a long-term vision and starting this program so that we don't have to borrow all the time. So we can make our own drugs that are unique and specific to juvenile dermatomyositis. So all of you wonder about this. What causes myositis? If we know the cause, we can find a treatment. We all know we take hepatitis B vaccine, so you are protected from hepatitis B infection. We know what causes hepatitis B. Likewise, there are monogenic genetic diseases where a single gene defect causes the disease. You replace with a better gene because you know what the gene is. In juvenile dermatomyositis, is more complex. These are called multigenic diseases. These are not caused by one gene, not caused by one mutation. There are multiple small genetic defects over probably half a dozen genes. So again, CureJM is looking at this genomics using the latest genomic technology. What are the genes that are causing this? So again, it is a long-term vision. So for us to understand how to develop drugs, we need to understand what goes on within the skeletal muscle. I know in here we have skin skeletal muscle involvement. I'll just give the skeletal muscle as an example. This is the beautiful normal skeletal muscle, so there is no problems. On the other hand, if you look at this, it's clear there is something drastically abnormal here. The abnormality is infiltrating immune cells. There are quite a few of them there. So what is the simplest thing that we can do? Try and get rid of them. Somehow everything else will become normal if you remove those cells. That has been the approach until now, not only for myositis, even for other diseases. So there very little investment was made on the muscle itself, the target organ of damage. So I'm not going to go through the entire pathway. I'll just summarize the top portion. That's what both Dr. Ryder and Dr. Agarwal have mentioned. These are extremely important pathways. They already proven if you intervene one of those lines, you can actually improve the quality of life for arthritis patients. So that's the same approach. But what we forgot is the bottom one, the muscle itself. So I'll show you some of the examples. The reason is simple. Often if you carefully study the muscle, you don't have inflammation, but you have a lot of weakness. When there is more inflammation, if the inflammation is the one that is causing everything, then you should see more weakness. That's not the case. So often confusing. It doesn't mean inflammation is not important. It's extremely important. But there is a lot other goes on within the muscle that we need to be focusing. So this is the model in which you have muscle that for whatever reason is leaking out stuff. One of the best things that we can see is the serum creatine kinase. It shoots up. But that's the only thing that we measure. There are a lot of other things get leaked out, what we call immunologically, they are called danger associated molecular patterns. That means they alert the immune system. Here there is a problem. This muscle is leaking. So because of this danger signal, all the immune cells arrive at this place. But many of them, when they come in, they damage the muscle. They also remove the dead and dying fibers. That's their purpose. So even to remove all of them, we need to be extremely careful. So what we have today is the blunt instruments that we all use, the prednisone, other immunosuppressive drugs. Now, all the latest drugs are also focused on, again, that component. So you heard many of these drugs just in the last two discussions. We need to have a different approach. Both Trish and Travis in Dr. Jim Iglesias' lab are now focusing on the bottom portion, and she will explain how we start the drug discovery with the muscle itself. So as you can see, every drug here, they are targeting to one of those pathways, none targeting the muscle. So it's extremely important. How do you target? It all depends on the success of the next presentation. So I'm going to stop it here. I just want to keep it simple so that Trish can explain the entire process of early drug discovery that they are starting for JDM.
Well, that's no pressure. I come bearing props. Thank you. So I'm Trish Dranchuk. I'm a senior research scientist in the Assay Development and Screening Technology Laboratory in the Department of preclinical innovation at NCATS, at NIH. We build acronym after acronym. And so today I just want to take a very few minutes to discuss how we partner with disease foundations like yourself. We are working with you actively. Dr. Kinder spoke this morning. He is your postdoc. And how we try to speed therapeutic discovery. Lisa mentioned earlier how many years it takes to get some of these drugs to trial. We're trying to make that more efficient. So some of these slides are slightly redundant from this morning, but I think there's some points that really deserve to be reiterated and explained a little bit more. So at NCATS, our mission is to catalyze the generation of novel methods and technologies, take those cutting-edge technologies that are out there, and incorporate them to enhance the development, testing, and implementation of new therapies and diagnostics across a wide range of rare and neglected disorders. So our group, part of NCATS, we're very collaborative, both in NIH and outside. So we have access to all the different resources at NCATS, which are pretty extensive, ranging from automation, those robots that Travis showed, analytical chemistry. We can actually synthesize molecules in-house, informatics, compound management, and then our new core facilities of siRNA screening and stem cell work. We also work with intramural labs throughout NIH as well as other government agencies, including the FDA and NIST. We work with a lot of extramural labs, universities around the world, as well as small biotech and pharma companies. And then most importantly, we work with the foundations and patient advocacy groups like yourself. So what our lab does is in our name. We develop those assays, those small biological tests that recapitulate some component of a genetic disease that we can quantitate in a very efficient and effective manner, leveraging the knowledge from the disease experts, including the patients. And we really want to keep you guys involved at every step. We don't view this as like a unidirectional process where we know the best and we're going to tell you. We really want to work as a network. We want to keep you involved the whole time. We want to feed information back to you and get feedback to us. And that's from the clinicians, from the patients, from the basic research scientists, everybody. And then we're going to apply our expertise in the analysis and development of progression strategies to evaluate very large, and when I say large, I'm talking 500,000 small molecules at a given time in our diversity chemical collections. And then our lab is a little different than most labs in NIH. We actually work with this postdoctoral model where the foundation supports a postdoc who's the champion for their disease, and this really serves two purposes. These guys are going to maintain the project momentum. They're going to be the hands on the bench every single day. They're going to be the ones responding to the emails and communicating with you with teleconference calls. But this is also a training opportunity for these young scientists. So hopefully they're developing a passion for your disease, your pathophysiology. So when they go on into their next stage of their career, whether that's in pharma, biotech, academia, wherever, they're going to take that interest and hopefully implement it in their new careers and help enlarge your study group. And as you can see, we've worked with a number of different foundations with this model for a number of different years. Okay, this is a slide I like to show in all my talks. I'm sure you've all seen the translational pipeline here to the left in some form before. It's always shown as this very linear process, starting with a disease or a pathophysiology, moving into assay development, large-scale screening, into lead optimization, and then clinical trials. And they always assign some arbitrary time point for how long each stage takes. And I'm going to tell you from working in this field for about a decade now, that's completely not true. Sorry. To the right here is a figure I actually took from a publication from a former postdoc in our lab working on Parkinson's disease with Michael J. Fox Foundation. And I really like this figure because it shows how cyclic and iterative a process screening and identification of new lead molecules is. We're learning more about the assays, the limitations of what we're screening, more about the biology of the disease. New technologies are becoming available at each step, and we want to feed that back into the previous steps as well as the current step of each step of this process. And at the same time, we're identifying new lead molecules, we're identifying novel chemotypes, and we're moving those forward and trying to validate them, but at the same time, we have backup plans coming into the pipeline through the whole process. So in our group, our program deliverables are we're going to work to develop a screening as well as follow-up strategies for anything we identify. We're going to validate and optimize assays. We're going to validate and confirm drug target and pathways, and then hopefully identify some active compounds or chemotypes. So how do we do this in an efficient and fast way so we can speed up that 10-year process? This is where my props come in. We're going to pass these around. This is what Travis and I work in every day. And Travis is going to need new glasses by the time he's done in our lab. So I'm sure all of you are familiar with the 96 well plate from your days in biology class. So for a few cc's of reagent, few mils of cells, you can get 96 individual experiments. We're going to put that on steroids and basically take it to 1,536 individual experiments for the same amount of reagents, the same number of cells, and the same amount of time, if not faster, because we have robotics that do this for us. And so you can see each reaction takes about 1/10 of an eyedropper volume, and that can be quantitated almost instantaneously on some of our readers. In addition, we can include 128 individual controls on every single plate we're running. So when we go to the robot, we lose some control. We have to trust that the robot is doing exactly what we told it to do. And anybody who's tried to deal with an iPhone or any technology, you know that there's limitations to that. So we want to be able to confirm very rapidly, yes, no, in real time, did this plate of experiments work? And that's what these 128 controls allow us to do. However, as we work in these very tiny wells, we have to remember we're moving further away from the biology of interest. We're not going to put a patient, we're not going to put a mouse, as even a C. elegans worm model into one of these wells. But what we can do is a patient cell, a model system, enzymes, protein complexes. All of these can be very easily quantitated with fluorescence or luminescence output. However, at the same time that we're working with these high throughput assays, we have to remember to be designing follow-up assays that get us back to the biology of interest. So we're not just developing one or maybe two assays for you, but we're going to develop a whole portfolio. Travis, I hope you're ready. We'll buy more coffee. So in our lab, we've had a lot of success with various foundation-supported projects in a number of different strategies. So as Raju mentioned, when you have a single gene of interest, you know genetic mutation in a single gene, we can easily put a reporter system right into that locus, especially with the advent of genome editing, CRISPR, Cas9, et cetera. And what that allows us to do is make that cell glow like a firefly, or fluoresce green, or red, or whatever it may be, that can very easily be measured for up or down signal. However, when the phenotype is more complex, polygenic, involves a lot of different mutations, we can open it up to a phenotypic cell-based screening. And Travis showed a little bit of this results this morning. But we can use high-content microscopy to image every single cell in every one of those wells on every single plate and do hundreds of those plates every single day. And this allows us to look for interactions between proteins, colocalization, cellular stress, and even cytotoxicity, all in a single readout. And then another effort in our lab is on the neglected disease side, which is part of my work as well, looking for parasite-based viability assays. So we're looking to kill some parasites from third world country diseases like malaria and osteoporosis. So this is a slide Travis presented this morning, but when you work with us at NCATS and we do this high throughput screening through hundreds of thousands of molecules, something that's unique to us is we add this quantitative element. So every single small molecule we test in every single assay gets tested across a concentration titration, from a very high concentration to a very low concentration. We live by the adage that the only difference between a drug and a poison is the dose at which you give it. And so to know that right off the bat is really a very powerful tool. So we use these robots. They run 7 days a week. They're down for about 3 hours a week to change on libraries and assays. But it's been quoted that the robots run 1 week. They can do the work of 10 researchers, what it would take them 10 months to do. So that's how we can really crank through a lot of these results. We obviously have a huge informatics team that is taking the results off the robot as it's being generated and immediately processing it with these concentration response curves that give you the pharmacology of every single compound in first pass. So we not only tell you, yes, this compound has an effect, we tell you how potent it is and how efficacious it is. and you incorporate counter screens with it, you can immediately eliminate those cytotoxic and those off-target effects and really help you rapidly identify your best molecules. And as I said, we have a ton of libraries at our division. We have over 10,000 pharmacologically annotated compounds, and we have every FDA-approved drug. We're trying to include those that have been approved in Europe and Asia as well as therapies in veterinary medicine. That would be our repurposing efforts. So obviously, if a drug's been approved for something else, they've already done the toxicity, we can speed it up in a new novel repurposed study. We also have hundreds of thousands of unique diversity collections. So these are molecules synthesized by our chemists, commercially available, in extramural labs that are supplied to us. And we have complex mixtures, including natural products. Nature does things the best, that we can also put through every one of our assays. My final slide is a case study with the Charcot-Marie Tooth Association. This is a foundation we've worked with the longest. We started in 2008 with them. They actually funded 2 postdocs in our lab over the course of this time scale. And you can see the different milestones and deliverables across the time scale. We developed a number of different assays, and part of that was that there was a lot of new technology coming into existence, including genome editing, through the course of this study. We had a number of papers. We did identify a number of drugs that are currently in lead validation and follow-up in animal studies right now. The publications we put out were for the scientific community as well as the patient community. And one of our greatest successes, one of the actual assays we developed in our lab, was licensed by Sanofi Genzyme, who's now putting it through their 1 million compound collection. So hopefully expanding those therapeutic leads even more. And with that, I'll thank the ADST team, and I guess we can open it for questions for everybody.
Thank you, Trish. Appreciate that. And thank you, Dr. Agarwal, Dr. Raju, and Dr. Ryder for your presentations. Excellent. We have a little over 15 minutes for questions and answers. So I see a question here, and I'm thinking maybe if we can come up here in just a couple chairs in our panel, that would be ideal. So just pull a chair. We'll have 4 chairs across here. Okay, great. Thank you.
So I think there's always sort of a tug of war between the recruitment and the complexity of the protocol. Okay, so that's what you're talking about. The protocol is so complicated and so ifs and buts that the patients are not able to get in. So in science, the difficulty is because, and also more so in rare disease, if you're doing a small study and you don't have a uniform population, then the results become meaningless. So we always try to make a good balance between promoting recruitment by making it simpler, but yet not putting in too many variables in a small study to kill the study. So that's always a tug of war. It's not about surveying the patients or anything. It's about keeping the science that will be valid for other people to review. I mean, even if the results are positive, if some reviewers look at it, it says, that's confounded. You're including 3 different types of patient, and each category is 5 patients, and we're not going to believe this result. Then it kills the study. So I'm going to have, yeah.
So, and another point on that also is about safety. So, like, if you're on certain treatment, then it might be unsafe to give, like, you can't take several immunosuppressives at the same time, or there might be some that are inhibiting the same pathway. So that's why they have to come off. And sometimes, frankly, we do advise companies, these are sometimes their designs, not the investigators' designs. And sometimes we do advise them not to design them a certain way, but they insist for whatever reason that they think that those criteria are important. Of course, they are the ones who have discussions with the FDA before they start these trials, and we're not privy to those discussions. So, we try to advise them that might inhibit their recruitment, but they insist on continuing with that design.
So I just want to make one more point. So recently, there was a drug company I was working with, and I told them we need to cover both dermatomyositis and polymyositis, and we have done combined trials before, and our outcome measures allow us to do those combined trials. So they got convinced, or we were able to convince them that when they went to FDA shut them down. And now they're only doing trial in DM, which is okay, but it would have been for PM patient, now they don't have the trial, So a lot of times things are not really in our control, and FDA has their own thinking. And to me, sometimes I feel that FDA don't have an expert panel in these diseases, and they don't consult us. FDA almost never consult us, to be honest, on these things. So a lot of times they're making decision based on who's really evaluating at that time point. So I've seen both ways. They've told us, why don't you do PM as well? And I've seen we go to them with PM and say, why don't you only don't you do DM only? So there's always been some problems because of lack of knowledge as well.
First of all, we do widely advertise the studies that we have, including I'll post on Pediatric Rheumatology listserver. We're at the American College of Rheumatology meeting. The same posters that hung up here, we're planning to bring them over to those meetings. We go to a lot of different meetings, give a lot of different talks, try to keep people aware of these things. But actually, in recent years, I will tell you honestly that it's you, the patients, that are getting these people to get into these studies. I'm getting more referrals from patients than busy doctors. And it's really important for you to, and I know some of you are really out there on Facebook and other forums, communicating with patients, encouraging them to get over to GW for a trial or NIH for a study. I know that because I get the referrals directly, and I know that you've sent them. And it's much appreciated. That's how the patients are getting to us. So you're making a big difference.
One good thing is if you just Google up the disease and treatments, clinicaltrial.gov shows up. Every single trial done anywhere is listed there. And it generally comes up at the top. We don't have to go to 25 pages down to see.
So one more point I want to make is what Lisa was saying is that, so busy docs, when you go to see a busy doc, they are the worst advocate of telling them to look into a clinical trial. Okay, so I think that is a real problem. They don't, even for a disease where we don't have any FDA approved treatment, they are not going to tell patients, oh, why don't you look into, there are options for you, but you can also look into clinical trial. That never happens. It only happens when a patient asks, can I go to a clinical trial, or when they're exhausted other options. By that time, they have had so much of damage and other problems that it's tough to get into a trial. So it's a real problem. I don't know how to.
Tertiary centers, individual rheumatologists wouldn't do it, but if you're at a tertiary care center,
even a tertiary care center, it's an individual rheumatologist who's operating at the time when they're seeing you. So it doesn't translate. The first time when you see them, they will never, even after 2 years, 3 years, once everything is kind of done, they say, okay, why don't you consider a clinical trial? So clinical trials are currently considered at the bottom option, as the last option, which I think is wrong. I think a lot of time, clinical trials should be considered as may not be the first, but the second option.
And I think in part, Kevin, you answered your own question that there are some things that CureJM can do through our chapters, through our website, through Facebook, through Twitter, through more, even stronger communication about the importance of enrolling in these trials. I think we've done a lot of this in recent years, promoting trials, but successfully enrolling these trials, and especially with young children, is extremely difficult. So we just need to get out there as much as we possibly can and beat this drum. Yep. Yeah. Let me repeat that question if I could for a moment, so we capture it on the camera. So the question is relating to IVIG trial, and are there other options that could involve younger children in subcutaneous work.
So there are two studies I'm involved with subcutaneous IgE. One of them is taking patients who are better after getting IVIG. So that study is basically saying, once you've done 6 months of IVIG and doctors have gotten you better, can you go to a simpler regimen so that you maintain your being good and not flare up because you couldn't continue the IVIG? So that's one of the study. The other study that I'm trying to get on board nowadays is to get a sub-Q IG to begin with. So you have active disease, moderate to severe active disease, and you treat with sub-Q IG, and it will be very beneficial for patients and younger kids for the same reason. But the study will first be done in adults, and then will be looked at in a smaller population with juvenile. And we hope that then once you have looked in juvenile, they will also get the permission.
Okay, so that being said, how far along would you have to be tapered off of your other meds? Are you just talking just IVIG, say they're on cell set like other meds too? Is that
Generally, we allow one steroid, a limited dose of steroid, generally it's about 20 milligrams, and another one additional immune suppressive drug to be on board.
I don't know how many of you have seen the series on Discovery Channel that was out last summer called First in Human. I really highly recommend you download that and get it and watch it. It's quite an inspirational series. It was filmed at the NIH Clinical Center in Bethesda, Maryland, and it's about really innovative new treatments that have revolutionized treatment of certain diseases like cancer immunotherapy, bone marrow transplant for immunodeficiency, and other such therapies. And really the heroes of the story are the patients that took the risk to come into these very novel treatment trials and really advance not only curing their own conditions, but taking tremendous risks to advance the science and the field and develop new medicines that revolutionize these disease treatments. So I found it a very, very inspirational series, and I think you will find it too. And I highly recommend referring other people to it as well.
Okay, I think we have time for 2 or maybe 3 more questions.
First of all, I'd like to thank you doctors for all the work you're doing. It's great. Just have one simple question. Who the hell names these drugs?
Try that one, Raju.
So there is a naming system. So the main idea is not so that people don't confuse with one to the other, so that you don't have the wrong prescription. So everything has to go through this nomenclature committee that looks at every, and they also came up with a weird way. Suppose if it is take a biologic, if it is coming from only all human, it has a different name. Now what we did, we genetically engineered tiny bit portion of the antibody from mouse, and the rest is from human. They have a different name. So they have a deeper meaning for each of those, but they're extremely complex. The idea is simple, so that if you just name it randomly, there is a confusion after some time. Some misprescriptions, labelings.
Oh, question in the back. Did we have a question? Yes.
I just have a comment, and I think we touches a little bit of this question and the trial, but I'm a mom. I don't have a background on science, biology, and when I go to doctors, I'm educating myself. I'm here to educate, to learn those names. When the doctor talks to me, I know what we're talking about. But I think on the trial part, even if I click the trial.com, I don't understand how to do this. And I would like to learn more. And I think it's a gap on that between parents trying to do the right thing, trying to educate ourselves, and find those busy doctors to tell me what to do, and us as a parent. So I think maybe what can I do as a mom to help you? You know, I know how the government works sometimes, FDA, you know, the NIA, I work for the government. Even if I have to request as a mom, as a U.S. citizen, I have the right to ask. So what can I do to help you to get everything that you need? So I want to help because I want to help my daughter. So anything that I can do, tell me, and I will do it. I will write any letter. I will touch any door. But tell me, and I'll help you.
We'll take that one back and do it. Right. Sherry, you had a question.
Yeah, maybe I should use the microphone so you don't have to do that. So I had a question about the adult patients when the JM adult patients when they get into the trials that are for the DM patients. Since some of those are going to be refractory patients, are they going to skew the results of the trial? That's one question. And then, I mean, for us, it's great because we want our patients in there, right? So don't get me wrong, and don't take us out of it. But then I talked to Octapharma at our last conference last year, and we had this ongoing discussion because they thought they could include us in the trial. But then they did decide not to, and that was kind of a concern. But they were also worried about, IVIG is approved for us. Well, not approved. We're able to get it through our insurance company because there's enough proof that it works. And their concern also was that, well, what if they did a trial and it showed that it wasn't effective and then we couldn't get IVIG anymore? So that was another kind of interesting point. But yeah, just wanted to get your thoughts on that. Both of those issues.
Okay. You want to?
Sorry, I was going to.
I can talk.
Got to go around this way.
Yeah. So one of the second question that you have is sometimes the company has their financial models, their business side of it. So a lot of times science makes sense, but business doesn't. So sometimes they shut us down, but most of the time science triumph. So I think and that was one of the situations where they thought that the business side didn't make sense. But I'll tell you, even the science side didn't make sense to include juvenile dermatomyositis, and I was part of this discussion in the panel where to include juvenile dermatomyositis versus not. And it boils down to the number game. So when we actually look at the number, if you're only going to enroll 90 patients and you expect about 20, 30 of them would be juvenile dermatomyositis, adult juvenile dermatomyositis, and then you do an analysis that 70 patients, 60 patients may not be enough to give you the result that you want. That's what kills it. If you were doing 200 patient study, then I always push for adult juvenile dermatomyositis to be here in most cases, but sometimes I tell them not to. Because when the numbers doesn't make sense, then you're going to kill the whole drug, even for adult. So that's why in some trials you will see that juvenile will come in, and some you will see they won't come in for the reason that what does the analysis show? That if given that it's a harder disease in adult juvenile dermatomyositis, let's say those patients don't do well, would we still have enough results, positive results or not? And if the answer is no, then I would say not include them. Because at least you get this one step forward to get in adults, and then we can do the next step. That's sometimes the better approach.
So you would have separated them out and not put them all in
Yes.
dermatomyositis. Okay.
If the number of the patients that we are studying is small.
Okay.
Well, I'd like to disagree because I have to say that, because I've been in meetings too recently with the FDA, and frankly, for orphan disease, we don't have any teeth to get pediatric studies done. We're not like adult rheumatoid arthritis where pediatric studies will be done for JIA, because adult RA is big enough. It's not an orphan disease. For orphan disease, they're going to do one study. And if you're not in it, you aren't going to be labeled. So I really think we need to be in all these studies. JDM, if we don't have a JDM specific trial, we should try to get in on whatever study is getting done. No matter how big it is. I don't really care how big it is.
When I've talked talked to some of these companies, I got the feeling that they weren't going to separate it out. Like what you were saying probably makes more sense, but I got the feeling they were just going to, right, I agree. But I just got the feeling when I was talking to them that, yeah, they're accepting, they're just going to kind of clump them together. They're like, oh, well, they have juvenile myositis, and so they're going to be, what? It would have to be labeled separate. They would not combine the results.
They'll combine the results, but then when they make the label, it will say adult dermatomyositis, polymyositis, necrotizing myopathy, overlap myositis, juvenile dermatomyositis. You'll be in the label.
So the talks that goes behind the scene that you not know is what happens is if there is a 100-patient study, and even though they want to combine everybody, the idea is if juvenile dermatomyositis patients don't do well, okay, let's say how many patients were we expecting of that adult juvenile, maybe 20. Would 80 patients be enough to show us positive result or not? If the answer of that is no, then that kills the juvenile dermatomyositis entry. If the answer to that is yes, we'll still have enough power to show the positive result, then I want to push them in for the reason that Lisa was saying. Yeah, it's a complicated,
I don't see why they're going to be more refractory than many of the adult patients who come into trials. And if companies are worried, they could say, okay, we'll take them if they have disease no more than 10 years. If you got disease at age 16 and you're now 18 and eligible for a trial, why should you be left out?
That's generally the recommendation that we make, that make a disease duration shorter. So you're not having, yeah.
We have time for one more question. Yes, ma'am.
The rituximab now, that knocks out the immune system for 6 months. It takes out the B cells for 6 months, so you're stuck with it for 6 months. And the newer drugs are targeting specific things in the pathway. Is there more control on how much they take out the immune system on that? And is it possible to regulate it? In other words, not take it down fully, just deregulate it down to something that's much more healthy?
Yeah, I think it's Raju. You probably want to answer that.
No,
Or
you can
you're
try.
You're the B cell. You're the basic scientist. I'm going to give the hard question to Raju. It's complicated. That's why I gave it to Raju.
It's extremely complicated, partly because immune system and immune response itself is a dynamic. So sometimes if you take cytokines, for example, interleukin-1, if I take my sample in the morning, afternoon, and evening, you will see the levels. They look as if they are collected from completely different people. That means my immune system is the way it is in the morning, the same thing is not in the afternoon. So controlling immune system in a measurable way is extremely hard because we are all the time exposed to various stimulants every single minute. So that means the system is going up and down, still maintaining the so-called normality of it. So I think it is a very hard task, but relativity, so there are 5 or 6 B cell, like rituximab-like drugs. The efficacy of each of those are slightly different. So we can only select which one is better based on their efficacy, as opposed to controlling the entire system is hard.
Those drugs that are targeting, do they last 6 months like the rituximab? Or are they something that if tried and you find that they're not working well or you need to move off of them, does it last that 6 months?
So yes, it's another complex question, partly because what happens is the average lifespan of an immunoglobulin is around 3 to 4 weeks. That means if there are different subtypes of them, IgG 1, 2, 3, 4, each of them have actually different half-life. Depending on if the drug is IgG4, it has a different half-life. Drug is IgG1, it has a different half-life. So what goes inside depends on the nature of the Fc portion of the immunoglobulin itself. So, and a lot of the time people try to look, when they design these drugs, they want to minimize, because these so-called Fc portions, they react with 5, 6 other things. They want to have minimal reactivity so that you don't have the just antibody induced problems, not the target problems. So it's all sometimes it looks like it is not truly real science. Partly it is mostly kind of an art to a certain extent, balancing things.
Just one positive thing. I mean, rituximab actually lasts a variable amount of time in different patients. Some patients don't knock all their B cells out and it doesn't work very well. Other patients knock them out. It could be a year or 18 months or longer. Some people it lasts 6 months. But the positive side of that is that if it works, it kind of resets things for a period of time, and then people are able to taper down on a number of other treatments. And this has some advantage in some patients, really. We've seen people get off treatment in that period of time.
Well, I want to thank you all for your rapt attention. I want to thank our panelists for your extraordinary presentations. Thank you. I want to thank all of you because your ongoing support of CureJM, both emotionally and financially, have made this kind of work possible. Especially Marge Coffey in the back of the room, who has been our champion funder for. I know, Marge, you're like, never want to see that credit, but for a lot of our work that's being done in drug discovery work right now is due to her tremendous support. So thank you, Marge. We wanted to get you up on the panel, but everybody needs to know you refused. Right. So thank you all. I think we have dinner at 6, which is, I'm sorry? Yes, dinner at 6. It is in the ballroom on the first level where we were this morning. And we'll look forward to seeing all of you there. Great. Thank you.